Healthy kidneys filter about a half cup of blood every minute, removing wastes and extra water to make urine. The urine flows from the kidneys to the bladder through two thin tubes of muscle called ureters, one on each side of your bladder. Your bladder stores urine. But what is renal clearance? Well, plasma clearance or renal clearance is defined as the amount of plasma that is cleared off a substance in a given unit of time. It is also known as renal clearance. It is based on Fick principle. So, formula to calculate clearance value is that concentration of the substance in urine into volume of urine flow divided by concentration of the substance in plasma. Unit of concentration of the substance in urine is milligram per milliliters. Unit of volume of urine is milliliter per minute, while unit of concentration of the substance in plasma is milligram per milliliters. So, unit of clearance is milliliter per minute. Now, let's understand the effect of filtration, reabsorption, and secretion on this clearance. If volume of urine flow is 1.2, concentration of the substance in plasma is 1.5, but concentration of the substance in urine is 30. So it means the substance is filtered, reabsorbed, but not secreted. Putting the values in equation. So the clearance is 24. Now, if volume of urine flow is 1.2, concentration of the substance in plasma is 1.5, but concentration of the substance in urine is 55. So it means the substance is filtered, but not reabsorbed and secreted. Putting the values in equation. So the clearance is 44. Now if volume of urine flow is 1.2, concentration of the substance in plasma is 1.5, but concentration of the substance in urine is 80. So it means the substance is filtered and secreted, but not reabsorbed. Putting the values in equation. So the clearance is 64. It means clearance is maximum if substance is filtered and secreted, but minimum if substance is filtered and reabsorbed, but not secreted. Determination of clearance value for certain substances helps in assessing the renal functions, such as renal blood flow and glomerular filtration rate. Value of factors, such as concentration of the substance in urine, volume of urine excreted, and concentration of the substance in blood, is required to determine the plasma clearance of a particular substance. Urea clearance test is a clinical test to assess renal function by using clearance of urea from plasma by kidney every minute. This test requires a blood sample to determine urea level in blood and 2 urine sample collected at 1 hour interval to determine the urea cleared by kidneys into urine. Normal value of urea clearance is 70 milliliter per minute. Urea is a waste product formed during protein metabolism and excreted in urine. So, determination of urea clearance forms a specific test to assess renal function. Inulin clearance can be used to estimate glomerular filtration rate. If a substance is freely filtered, means filtered as freely as water, and is not reabsorbed, or secreted by the renal tubules, then the rate at which that substance is excreted in the urine is equal to the filtration rate of the substance by the kidneys. The glomerular filtration rate, therefore, can be calculated as the clearance of the substance. Taking an example, a substance that fits these criteria is inulin, which is a polysaccharide molecule with a molecular weight of about 5,200. Inulin, which is not produced in the body, is found in the roots of certain plants and must be administered intravenously to a patient to measure glomerular filtration rate. In this example, the plasma concentration is 1 milligram per milliliters. Urine concentration is 125 milligram per milliliters, and urine flow rate is 1 milliliter per minute. Putting the values in equation, then inulin clearance is calculated as the urine excretion rate of inulin divided by the plasma concentration, which yields a value of 125 milliliter per minute. Thus, 125 milliliters of plasma flowing through the kidneys must be filtered to deliver the inulin that appears in the urine. Inulin is not the only substance that can be used for determining glomerular filtration rate. Other substances that have been used clinically to estimate glomerular filtration rate include radioactive iothalamate and creatinine. Now, clearance can be used to estimate renal plasma flow. Theoretically, if a substance is completely cleared from the plasma, means it is filtered and secreted, but not reabsorbed, the clearance rate of that substance is equals to the total renal plasma flow. In other words, the amount of the substance delivered to the kidneys in the blood would be equal to the amount excreted in the urine. Thus, renal plasma flow could be calculated using this equation. Renal plasma flow equals to clearance, because the glomerular filtration rate is only about 20% of the total plasma flow. A substance that is completely cleared from the plasma must be excreted by tubular secretion, as well as glomerular filtration. There is no known substance that is completely cleared by the kidneys. One substance, however, para-aminohippuric acid, is about 90% cleared from the plasma. Let's take an example. Plasma concentration of para-aminohippuric acid is 0.01 milligram per milliliter. Urine concentration is 5.85 milligram per milliliter, while volume of urine is 1 milliliter per minute. Putting the values in the equation, so renal plasma flow is 585 milliliter per minute. Therefore, the clearance of para-aminohippuric acid can be used as an approximation of renal plasma flow. To be more accurate, one can correct for the percentage of para-aminohippuric acid that is still in the blood when it leaves the kidneys. The percentage of para-aminohippuric acid removed from the blood is known as the extraction ratio of para-aminohippuric acid, and averages about 90% in normal kidneys. In diseased kidneys, this extraction ratio may be reduced because of inability of damaged tubules to secrete para-aminohippuric acid into the tubular fluid filtration fraction is calculated from glomerular filtration rate divided by renal plasma flow, which is the fraction of plasma that filters through the glomerular membrane. One must first know the renal plasma flow and the glomerular filtration rate. If renal plasma flow is 650 milliliter per minute and glomerular filtration rate is 125 milliliter per minute, so the filtration fraction is 0.19. So this is the importance of renal clearance. Through renal clearance, we can find fraction filtration. This was all about the renal clearance. Hope I have well explained the concept of renal clearance and how glomerular filtration, renal blood flow, and fraction filtration can be found using renal clearance.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.